Welcome to What's Next from AgResearch, a podcast exploring science answers to big questions. I'm Erin. I work at AgResearch, but I'm not a scientist. I do have lots of questions about our science, and I thought that you might too. In this episode, I'm asking if new technologies like precision fermentation are going to wipe out pastoral agriculture. One of the key parts of AgResearch's work is innovation, new ideas to solve problems, lessen negative impacts, or create value. Our scientists work right the way through the pasture-to-plate process, from cultivars of grass, the genetics of the livestock that eat that grass, the nutrition and welfare of those animals, as well as the quality and the quantity of the meat and milk they produce. And that's where I want to start today, with food. AgResearch has a team of really talented people exploring the ways to make the food produced by agriculture tastier, maybe more nutritious, and maybe even personalised. There's also work looking at different ways to produce food, that is, alternatives to pastoral farming for meat and dairy, like precision fermentation. So how real of a threat are they? To help me understand, I'm introducing you to a man who'd never seen sheep before coming to New Zealand and starting at AgResearch 28 years ago. Scott Knowles specialises in nutrition and biochemistry as a senior scientist at our site in Palmerston North. Hi, Scott.
Kia ora, Erin. Tēnā koe.
Let's start with the most pressing question. Do alternative proteins and lab-grown foods spell the end for our dairy industry?
It's a single answer no. All the alternatives are, in our context, is about and not or. It's really what can they bring to the table, both metaphorically and literally, that we're doing well, but we want more of, that we're doing in a certain way, but we want just a portion of it. It's how do you add new capabilities and new functionalities to foods that we're already good at producing. And so that the meat and the milk that we do so efficiently, they'll always be in high demand and they'll always be high quality whole foods. The alternatives are additives to that.
So good alternatives and not a replacement.
Oh, heavens no. Not there would be arguments around the world that people want it that way. And that's okay. You can have that point of view. But for our ecosystem as well as our economic models, we're going to keep farming for the foreseeable future. Within New Zealand, alternatives will always be a small production and a small internal market. Everything's for export. So we'll keep exporting what we do well. And if we can top that up with some high value functional ingredients produced by other methodologies, that's another revenue stream. It goes into different kinds of markets than our basic foods do.
Well, that's great. So now that we've got that sticking point out of the way, can we just go back a little bit? Can you tell me what precision fermentation actually is?
Well, precision fermentation is just one of alternative sources of foods. There's many ways to make foods that are not in a traditional growing way, but PF is really very interesting, and it's becoming more modern, though it's been around for years. So let me just say, at its basis, it's training microbes to grow in controlled conditions in which they can produce like little cell factories and produce functional, specialized compounds that we particularly want rather than what the bacteria wants. And so that by using some genetic engineering techniques, you can reprogram microbes, various kinds of microorganisms, to produce things that they otherwise wouldn't or couldn't, but are of benefit to us. Once that fermentation is done, you separate the microbes from the thing that they have made, and the microbes go back to their job in the micro farm, in a sense, and the products become, lactoferrin or beta-lactoglobulin whey's or caseins or other useful ingredients that we want to do things with. The microbes have just produced them as little cell factories. And so from a dairy point of view, we think of it as stainless steel cows.
Okay, right. That makes a bit of sense then. So this isn't, like you said, it's been going on for a while. This isn't unique to New Zealand. So why would we want to do it here?
The PF started decades ago in the 50s and really hit its stride in the 80s with the production of insulin. And so almost all of the world's supply for decades has been made by precision fermentation of various kinds of insulin, usually human insulin, rather than isolating it from pig pancreases. It's a much better way. And so since then, PF has been used around the world to make ingredients for food, to make medicines, to make vaccines. So some of the technology is just not new. It's not surprising. Why we would want to do it here is, one, for the revenue stream. If anything else we can do that makes a quality product that we can export is good news. And the second is to kind of have New Zealand take a position on these alternative foods and alternative methods. They're coming, they're growing in different parts of the world at different rates, but we as the scientists and as the food producers and as the country as a whole need to get in front of if not the arguments, get in front of the discussion and say, this is how we feel about it. This is how we're going to participate in it. This is how we're going to lead. And it's better to do that purposefully than just catch up or always being on the defensive. So in New Zealand, our resources are special. And one of the definitions of special is small, unfortunately. The things that we can do here, that we should do here, should play to our strengths. So we've got lots and lots of dairy agribusiness experience. And on top of that, and supporting it, is dairy science and dairy processing expertise. So we know how to make good dairy proteins, what good dairy proteins look like, what happens when they go wrong, what to put them in, how to use them. We can use that same kind of expertise, but with dairy proteins that have been produced in other ways. So, we are arguably in this hemisphere the world's dairymen. And we just want to have a different raw material that we can use our expertise on. So
what do we actually make it with? So talk to me about that.
Mm-hmm. I speak of microbial farms, or of these things growing. They don't grow on just air, though there are some folks in Finland doing that. They need a substrate. They need a culture medium.
So do you mean like a food source?
It's a food source that allows the bugs to grow, gives them the right kind of nutrition, so that then the programming that they've been giving can be churned through in little machinery to produce products. Usually in most precision fermentation around the world, that food source is sugars. Usually sugars that have been isolated from sugar beet in the northern hemisphere, from sugar cane in the southern hemisphere. You've probably done it in a high school experiment. If you feed sugar to yeasts or bacteria, they grow and foam and just go crazy. All right. So that's the basic way to do it. But from my and many people's point of view, sugar is a real food. Should we be taking sugar out of the mouths of us or whatever we want to use it for and feeding it to microbes? So one of the things that we're interested in at AgResearch and our science collaborators, and is an advantage for New Zealand, is using different bioresources as the food source. So take, for instance, an example, we've got lots of forestry waste. Forestry waste is full of lignocellulosic things. It's all the stuff that makes wood tough. Those are big molecules, but they are fundamentally made of sugars, and those sugars are connected to one another in complex ways that give the plant strength. If you can break those down, you make a sugar solution that you can feed bugs. So the connection there is taking waste, processing it in a way that makes it edible for microbes. The microbes do interesting things with it and give us products out the end.
So you're talking about making food from forestry waste. That's incredible.
Could be. That's why not? Why use an already good food material just to make another food when we've got plenty of bioresources here? And it's not limited to just forestry resource. Any plant-based material has these celluloses in it. And depending on how you enzymatically and chemically and physically process them, you can break it down into food for microbes. Microbes will grow on most anything. Just look at the back of your refrigerator.
That's so true.
It doesn't take long before they figure out how to eat what's available.
Okay. So will the outputs of things like this, if you're talking about dairy-like foods,
yes, will
they be nutritionally similar?
Yeah. That's the great thing about precision fermentation, is it's the only way to make bioidentical materials, because you're programming the microbes with genes from the species that makes the original. And that might be a dairy cow. It might be a beef animal. It might be a chicken egg. embryo. You're then telling the microbes, use your genetic machinery to kick out exactly the same thing that this gene does in the original animal. So you get bioidentical proteins or lipids or carbohydrates. And so are they edible? Well, of course. Are they the same? Well, yes, because they're bioidentical. I have a nutrition background, and I guess I'm classical or fundamentalist in it. And I believe in whole foods wherever possible. And there is a lot to be said for the way the components of foods through the eons of evolution have come together to create sums that are greater than the ingredients. So you look on the back of a pack of food and it has a breakdown of the protein, carbohydrates, blah, blah, blah. That's not how the food grew, and that's not how you metabolize it as a reductionist way. So while precision fermentation can make individual perfect ingredients, they need to be used in ways that are cognizant of the fact that it's not a whole food. So if you're making a casein or a beta-lac whey protein out of precision fermentation, that's great. It'll do its job, but it's not milk. It's a component of milk. You have to use it in a way that is appropriate for that subfraction. So whole foods will still always be the gold standard.
Okay, so again, we're not talking about a replacement. We're talking about an alternative and an extra thing. Mm-hmm. Mm-hmm. So how hard will it be to scale? Because when you're talking, I could see that happening in a lab and you making, a bowl of. A bucket full, yes. Yeah. Mm-hmm. So how hard is it to scale? And do we need to scale it up heaps or just a little bit?
Let's see. There's no there's no short answer. The general one is it depends on the product that you're making. If what you're making is very high value on a per kilogram basis, and the world doesn't need a whole lot of it, like insulin, then the scale is not too much of a problem. You can do it in fermentation vats on the orders of thousands of liters, and they bubble away like big vats of wine and can produce the right amounts that create value. But if your aim is to substitute for existing dairy, as in some of the Northern Hemisphere companies, Perfect Day is currently selling whey proteins to use in ice creams and some other things. Well, that then it's a commodity in the same way as our dried skim milk is a commodity. You have to make lots. So that scale for making a commodity product from precision fermentation is outside of New Zealand. We don't have the stainless steel. We don't have enough of the engineers and the other people to run the very large plants to do that. Our angle on this is useful, eco-sensitive raw materials to feed the bugs, and then targeting high-value niche proteins or other ingredients that can be done at manageable scale. I was talking to one of our lead scientists, David Hooks, who really is across this whole brief and knows it in and out, and has helped develop a great proposal to bring AgResearch further along on this journey of precision fermentation. And I was asking him, what do we have to do to make lactoferrin? Lactoferrin is a fantastic protein out of milk. It goes into infant formulas. It helps absorption of iron, and it's antimicrobial and antioxidant. The world supply of lactoferrin, can we do that in New Zealand? And he did some back of the envelope calculations and says that we can, in using fermenters of just a few thousand liters, create all the lactoferrin the world needs. So that would then substitute for all the hundreds of millions of liters of milk that have to be filtered to get the lactoferrin out now. So it's an example where it's a high-value protein. The world has a certain fixed requirement for it, and it is of a scale that New Zealand could lead in.
And does that mean then, if we're already using heaps of milk to get that protein out, does that mean perhaps we'd have less waste product as well? That milk could be used for other things? Yes,
the dairy industry is clever and would say they have no waste.
Right.
Because it's just lower value streams that have different purposes. When they're taking out the lactoferrin, what's left is still good quality, and it has wide range of uses. So it's not like you're burning all of the milk just to get this, which they refer to as pink gold.
Right. Okay. Well, that helps to quantify in my head how much it's sort of worth.
Go with $1 million a ton.
Okay.
So It's good stuff. And
it sounds like it.
When precision fermentation comes online and can make it more efficiently, then demand for it will go up. Right now, there's not enough of it around to supply all the needs that it might have.
Now, we've sort of mostly been talking about protein, but that's not the only thing that you could be using this for. Am I right?
That's right. That's right. Protein is the easiest example, and it's the ones that PF started with decades ago. And all organisms have to make and survive on more kinds of categories of compounds than just proteins. And so they make fats, a large range of lipids, and they make carbohydrates. Plants are particularly good at making carbohydrates because that's how they make their woodiness and their leaves and the rest of it. So there is also precision fermentation happening in the lipid area where people are training microbes to create the same fats that are in dairy milk. That is a much higher bar to deal with than the proteins. Protein, doable, fats hard. Then you move up into carbohydrates, and that becomes quite difficult because people will know that genes code for proteins, and the proteins then create enzymes that then do their machinations and create carbohydrates from that. And so it's a lot of steps going back to teach the microbe to make different carbohydrates than it wants to. So an example that I've spoken of at other times, and which I think captures the imagination of this, is something called human milk oligosaccharides. And these are complex carbohydrates that are in all milks, but are a predominant composition of human milk. And we're starting to be able to precision ferment these. That means that then we can have a supply of these HMOs to go into infant formulas. Cow milk has very little HMOs in it. Human milk has quite a lot. And so in order to humanize cow milk infant formulas, you want to keep adding things in to make it more human-like. And so PF to make HMOs is just a great win-win for everybody.
So that would make formula, infant formula, closer to what they would get if they were breastfed.
Absolutely. That's right, to humanize it. And the HMOs are just fascinating because we think of carbohydrates as sugar and gives you energy or whatever. Yes, they do. They're important for the infant's growing and energy, but their real brilliance is that think of them like fibers in your dietary cereal. They are there to feed the microbe population that is developing in the infant's gut. The infant starts out nearly sterile, and the microbes have to populate. And the way that the nature controls what populates and that the infant gut just doesn't go feral is by manipulating the feed for them. And so these HMOs are the perfect feed to grow the perfect bugs for the infant to start its life with. And if you don't have those, the infant is missing out on this microbial advantage that gets them started.
That is fascinating. I feel like we could probably talk about that all day.
Very good.
So we've established it's not just about protein. And I know that that's something that you feel really strongly about, that the animals that we produce in New Zealand are sort of ideally designed. They're not just a bag of protein. I'm sure that I've heard you say that before.
Part of that is a little bit of a philosophical and pedantic argument of reducing what we create here or anywhere where livestock and animals are grown to just a simple protein on the label of a package. That I hate the thinking that oversimplified thinking that New Zealand exports protein. Well, yes, we do, but no, what we really export is the complex, generous nutrition that is in milk and milk powder and in the whole meat and the other kinds of animal products that we export. So I've always felt that the poor farmer who puts his heart and soul into healthy livestock grazing around and then sees and hears that he and his animals are reduced to this idea, well, we're exporting protein. When really it's so much more than that. It's whole foods that giving you nutrients that you can't get other ways. It's a little philosophical. I think animal farming is okay for what we need to do. and it will always be part of New Zealand's economy. And my guess is that even in a long distant future, there will still be global demand for safe, high quality whole food materials.
So we've talked about precision fermentation, and that's really only one example of emerging foods, isn't it? Really, are there other things we're working on, Scott?
We have I'm throwing around the royal we, but this is between AgResearch and the Redet Institute, which is a center of research excellence dedicated to nutrition and protein, based in Palmerston North, have just co-appointed a chair of alternative proteins. So this is a person that has been brought in from overseas after a long couple of years of search in order to give impetus and headline and mana to a position we can take in proteins, or not proteins, but in alternative foods grown from cellular agriculture, where you're growing up cells in order to mimic meats. How much future do we have of that in New Zealand? There's a lot of much bigger organizations working on it, but we want to get out in front of the argument. We can't be on the back foot.
Innovation is all well and good, but without the stuff in the background, it's really hard to pull off. So in AgResearch's case, that's labs and fancy equipment, and it's the technology to run all of that and then store the volumes of data we generate. So it feels like a really good time to stop and talk technology. Ben Taylor is AgResearch's infrastructure and security manager and formerly enthusiastic gardener, slightly less so after that run-in with Legionnaire's disease last year, Ben?
Yeah. Thanks, Erin. Yes, if you've never heard of someone who's had Legionnaire's before, I can be that person. I was gloved up and wetted my potting mix down, just to be clear, but I've learnt that doing it on a really hot, windy day is also not advised. So there you go.
I'm very pleased that you survived and you're here to tell the tale, and also give me the rundown on some of the tech stuff, because science today in some parts looks very different than it used to. And one of those is how much computer power we have and how much we need. Can you tell me a bit about what's required to power AgResearch's work?
Absolutely. So traditionally, when we've been dealing with science challenges, we've been thinking it constrained to what you can do on your desktop or your laptop computer. And that can only solve problems of a certain scale. But we have much bigger challenges today with climate change and everything else we're trying to address here. And that requires a much bigger computer. So we've got what we call our AgResearch infrastructure, and that's equivalent to 250 laptops together. If you think that combined power is what we have on offer for our scientists. And with that power, that allows you to answer bigger questions and do things at scale.
Well, that sounds pretty great. So how much data can it generate, and what can you do with all of that?
Gotcha. Well, it doesn't generate data. Scientists generate data, but it can store around 2 petabytes of data at the moment. That's how much we have. And that's equivalent to about 1.5 million videos, a million hours of video if you're watching Netflix. And we generate around about 100 hours or 1.5 terabytes of data every single day.
That is a lot of data.
It is. It keeps me interested.
I bet it keeps you busy as well.
Yes.
Something that I guess anyone who's got a computer at home or even a television or a phone in their pocket knows is that technology is constantly changing and getting, well, we hope better. How do you stay on top of all that and know what the next big thing is and how much you're going to need?
It's a great question, and I try and work with people much smarter than me. So we're partnering with an organisation in New Zealand called NESI, which stands for New Zealand's e-science infrastructure, and they're thought leaders in the space. So they deal with what's leading edge in terms of science and innovation, and how do you solve these problems. And so they've got a great idea in the architectures and the systems that you need to kind of address these challenges in a way that'll work. So I'll lean heavily on their expertise, and we've worked with them in designing the systems we have today.
And so is it just the things that we have, the systems that we have inside AgResearch, or do we have access to other bits and pieces around to help us deal with all of this work and all of this data and all of this sort of stuff?
Yes, yeah, absolutely. So while this AgResearch infrastructure is part of the tools that we have. We also work with public cloud services such as Microsoft and Amazon. So where we see a particular value in a product they may have, it's perfect for us to try and experiment with new technologies to see if they fit the purpose for us.
And so with all this power, what does that mean that we can do?
It means we can do some really cool things. One thing I love that we have in this platform is we have a capability called a GPU capability. And for those of you who are gamers, you may know about graphics cards. We have a really big one called an NVIDIA A100. I won't get into the details, but that just lets us do some really cool AI, machine learning and deep learning kind of technologies or applications.
And I'm just going to stop you because this is agricultural. When we say AI in this sense, what we mean is artificial intelligence rather than artificial insemination, I'm assuming.
Correct, or I'm doing my job wrong. Yes, that's right. And that's where a lot of the innovation is happening. And we have real practical applications for this. I think, there's a lot of hype around AI and a lot of things happening, but we've found some real tangible ways to benefit the challenges we face today.
And what sort of things are those?
I think one that at least makes a lot of sense to me and resonates is using machine learning to identify weeds in a paddock. So we have a few projects where we might have a piece of technology scanning a paddock, and we can quickly identify what weeds are there and how we could probably treat them.
That sounds pretty clever and quite helpful if you're a farmer. You could possibly have that in your pocket.
Absolutely. I mean, personally, I wish I had one at home for my garden. So I guess other examples are we'd have to start referring to some of the smarter people at AgResearch that apply this technology to problems we face today.
Well, luckily, we have one of those people with us today. Sam Hitchman is a senior data scientist. He's got a background in physics and imaging, and he's also deep in parenting with 6-month-old twins. So if it sounds like he might be running on a very strong coffee, that's because he is. Sam, hi.
Hello.
Can you tell me what sort of thing you're working on that involves these, huge volumes of data and this processing that we can now do?
Yeah, So I work in a range of different areas. As Ben mentioned, we're very good at generating lots of data, but you actually have to be able to analyze, go through that, get some results out of that. And so I work with across a whole bunch of different science teams at AgResearch to try and automate some of their processing, so that they don't get stuck sort of manually going through images and annotating things. Let the Flash computer do that, so that they can go on to do more sciencey things.
So we're letting our smart people do the smart work and the computer do the boring, repetitive labor. That sounds like exactly what this stuff is for.
That's right. There's sort of an upfront cost to develop these models, but it's sort of a one-off, right? Instead of year on year, your scientists being locked into needing to do that analysis, let the computer do it. And
what kind of applications are you working on for this kind of tech?
One area that I've been quite active in over the last few years is we have had at AgResearch a rich history of CT scanning animals for their to understand their genetics and their anatomy. And so a CT scan is a really cool bit of kit used in the medical industry routinely. It's basically a spinning X-ray scanner that recreates your internal anatomy. And it catches everything. It captures the lungs, the muscle, the skeleton. And so for a long time, we've been using that to extract the lean meat yields of breeding animals so that we can choose the best ones for our next year's progeny. But with more and more advanced tools to do the analysis, we're moving on to other traits to do with animal welfare or methane output, all sorts of things. So
that sounds very, very fancy. We haven't got time to talk about the logistics of getting a sheep into a CT scanner. I think that we might have to save that for another episode. Talk to me more about, you said methane there. Obviously, that is it's a hot topic. It's something that we're working on in lots of science areas, it seems, across AgResearch. So tell me more about how the scanning can help with that.
So, we're obviously at AgResearch have been very interested in the methane emissions of animals for a long time, understand understanding the mechanics of that. And so the rumen microbiome is being analyzed all the time. And my contribution to that bit of work is that in our sheep that we've bred to be high and low methane emitters, what we see is that there's an anatomy change of the rumen. And so understanding that process, and then also being able to see how the rest of our flocks stack up against those known methane emissions is very interesting, but not feasible to do if you were to do it manually. And that's why having the horsepower and the expertise to do that automated analysis at AgResearch is really important. For one thing, I can run the new automated analysis over our back catalog of 20 years worth of 1,000+ animals a year, but also for going into the future. I can basically provide that additional analysis for no cost for people that are already putting their sheep through the scanner. So it's really nice, shore up the fundamental research and be able to provide that information to farmers.
That sounds like a really great combination of benefits, and for a lower cost for farmers, which is always good. Bottom line is always quite often top of mind. So that's really great. Thank you very much, Sam and Ben.
Thanks, Erin.
Thank you.
You've heard Scott, Ben and Sam talking about just some of the exciting innovations using new technologies. AgResearch works with forward-thinking partners on projects providing the solutions for tomorrow. If you'd like to partner with our researchers on your next venture, learn more and get in touch with us at agresearch.co.nz or click the link in our show notes.